Hey Beaklings, it's Mr. Boylan, and I would like to talk to you about enzymes today. So here we go. Okay, I had you do a little background reading before we talked today, and what it was about was the energy of activation barrier. Okay, for these reactants to become products right here, they have to get enough energy, okay, added to them that they can cross this barrier, right? For a reactant to become a product, there's a certain amount of energy that has to be invested in this case. So what an enzyme does is the enzyme actually lowers the amount of energy, okay, needed for this reaction to happen. It crushes that barrier down. Okay, it absolutely does not, again, it does not add energy to the system. It just lowers the amount of energy that's needed. Okay, and shown here as a purple little molecule. Okay, and generally they're proteins, but as some of you know, it could also be an RNA molecule. Here's another pictorial representation of that, and this is just showing some jumping beans, and they would have a very hard time getting across here, but the enzyme crushed the barrier down, so now it's the exact same reaction. Okay, they're going to become products, and it's much easier this time for that to happen. Okay, they didn't have to expel as much energy, right, to get up and over that barrier, because the enzyme works by lowering the energy of activation. And here you can see a graph that represents these reactions. Some students find this a little confusing because this graph doesn't show the rate, right? But what it does is it just shows that the reactants are going to become products, sure. But if you have an enzyme involved, well, that needed much less energy, okay, for this reaction to happen, and that would obviously increase the rate. And one thing that's also important about this is for you to understand that enzymes can be reused over and over and over again. They're just catalysts. They're biological catalysts. So once you use them, you can use them again and again and again. Okay. So as we go through here, enzymes are super, super specific. Okay. Remember, again, enzymes are proteins. Almost every enzyme is a protein. And proteins, if you recall, have the long chains of amino acids that make them up, right? And most times you're ended up with like a tertiary structure. Those interactions of those R groups of the amino acids, oh boy, do they form this ultra specific 3D shape. And this dictates what the enzyme shape will be, and that then creates this thing called an active site. This active site is very, very, very specifically shaped. Okay, that's kind of like the lock, and the substrate that fits inside the reactant here, this is kind of like the key that fits in, and theoretically just this one key should fit this lock, right? So it's actually more specific than that in a lot of ways. Induced fit is when they come together. The substrate binds in here, okay, to the active site. We call it the enzyme substrate complex, okay? And what happens is when they do bind, sometimes it gives it like a, I like to think of it as like a little hug, a little squeeze. Okay, it comes together and everything's held nice and tight in there. And some people actually talk about how this forms a kind of like a little microenvironment in here. It's almost like the environment that's going on between the bonds in here are different than what was out here or what was originally here. Okay, because this is a different little area in here now. Okay, that these things are reacting together. So here's another example of it, and hopefully we had a chance to play with some enzymes in class, and we probably played with invertase, okay, which is another, the common name for sucrase. So as you can see here, we have our enzyme, okay, we all know how to make this. We all break down just regular table sugar, okay, so what's happening here is this enzyme is going to break down table sugar. This is what you sprinkle on your cereal, right? Or put in my Earl Grey tea every day. So this is glucose and fructose, right? The glycosidic linkage here. So that is a disaccharide. Okay. So how do we break this down into monosaccharides? Well, that would take a long time and a lot of energy. So what happens is the enzyme, okay, again, sucrase or invertase, comes along and it hydrolyzes it. Of course, dehydration, you build, hydrolysis, you break, so here comes our water and to help facilitate the breaking of that covalent bond in between. Okay, and it's pretty interesting how these work. It's almost like I don't know if you know what these are. And we're getting pretty high tech here. These are paper clips. So let's just imagine that these two things are held together like this. Okay, so you can consider one of them like glucose and one of them fructose. Okay, so I have sucrose here. I have a disaccharide. And if I wanted to really pull them apart, wow. Okay, and I don't want to make my muscles work too hard because I don't want to scare you. But point is, this is tough for me to do, to break this bond, and that bond will be there a long time. In other words, there's a high energy of activation for this reaction, okay, for this catabolic reaction of breaking apart these monosaccharides. So what an enzyme kind of does, let's pretend my hands are kind of like the enzyme, okay, and the active site is on my palms here, okay, or in my fingertips. And what the enzyme really kind of does is it takes these monosaccharides, okay, which are connected into a disaccharide, and it takes this disaccharide and it kind of just gives it a little hug, induced fit. And if you notice here, instead of me having to pull them apart, what the enzyme does is it holds them in a specific configuration where they kind of just, they just kind of fall apart based on changing shape, changing configuration, maybe changing pH, changing the way the R groups are connected, maybe new kind of bonds are forming in between them. So the enzyme really doesn't do a whole lot. It just kind of reacts and holds things in a different configuration, and that really changed the energy, right, needed to hydrolyze, in this case, sucrose. So let's see here. The environment certainly affects enzyme activity. We've talked about this with proteins, right? So hopefully, if you remember those proteins, you know that the primary structure is just a long chain of amino acids. Okay, that's pretty tough. That's peptide bonds. They're held together. But when you get past secondary, is just pretty weak. Alpha helix, beta pleated sheets. When you get into tertiary, okay, now you have a 3D functional unit. But you remember that those can be pretty much denatured pretty easily, right? With some reasons here. So hopefully this is a review. Each enzyme has a unique environment where it works best. Okay, and temperature can speed up molecular motion, so more substrate bind, sure. But you all remember that, right? Sure, if this is the enzyme and this is the substrate, well, sure, every now and then, this is just like kinetic molecular theory. Oh yeah, they'll hit, okay, or something will deliver it here. This is in a membrane, and this guy will get. But point is, eventually, if I heat it up, these are going to go faster and faster and probably hit more often. But if I heat up too fast, these bonds in here, these are amino acids being held together by sometimes just weaker bonds, ionic in solution, or hydrophobic interactions, hydrophilic interactions, little H bonds in here, things like that. Whoa, the faster I go, this can denature, and all of a sudden my substrate can hit it, and nothing's going to happen, right? So heat can definitely denature a protein, okay? And shape dictates function, obviously, so if you lose that, you're in big trouble. Here you can see hot spring. I think this is morning glory, perhaps, from Yellowstone. But optimal temperatures, most of your enzymes, of course, work best at your body temperature, around 37.5 degrees Celsius, whereas hot spring bacteria, no, they need it much hotter, right? And if you took your enzyme and put it in there, it would denature. And if you put their enzymes inside of us, they probably wouldn't work well either, okay? And, of course, salinity, right? So we talked before, the concentration of salt can affect enzyme activity because of the ions, right? Whenever you have these ionic compounds, you have little charges in there, and if they start pulling on that tertiary structure, you might not have to lose much of it before the enzyme's non-functional. So the change in pH, of course, can affect it as well. Obviously, you have these little protons and things floating around in there, or lack thereof, and yeah, that'll change what happens. Optimal pH generally is about 6 to 8, so around 7 probably, which is a lot of our bodies. But some lakes have so much acid rain that, yeah, the aquatic organisms' enzymes lose their functionality. It's kind of sad, huh? Because of our pollutions and the stuff that we're putting in the atmosphere. It's a little confusing. You have cofactors. They're little inorganic helpers. They're kind of just like minerals. Okay. Sometimes enzymes need help. That's why it's so important to eat a healthy diet. If you eat normal food, if you eat like you're supposed to eat every day, you're fine. But you have to make sure that you get all these vitamins and minerals. Okay. And as you can see, some of these minerals here, they help with enzymes. The bigger ones are the vitamins, right? The coenzymes here. And as you can see here at the bottom, this enzyme might not really work very well on its own. oh, but if you have this vitamin or this coenzyme inside, it can actually help this active site change into something that's, there we go, that's perfect, and now it'll work. So some enzymes on their own don't really work until they get some help, okay? Comprende. Some human enzymes work best around pH 2. Where are they probably located? Well, low pH probably in the stomach, right? Okay. All right. Enzyme inhibitors. So whenever I hear the word inhibition, I think stop, right? An inhibitor is a chemical that interferes with enzymatic activity, and it stops the enzyme from working. So when covalent bonds form, an inhibitor may be permanently in there, right, in the active site or at a different spot, and you might ruin that enzyme forever, okay? But sometimes inhibitors might just weakly bond to it with little H bonds and intermolecular forces, okay, van der Waals and things like that, and yeah, they'll fall back out. So that's kind of like the enzyme can oscillate on and off in a way. But there's two types of inhibitors that we'll cover here, and it is a competitive inhibitor and a non-competitive inhibitor. So the competitive inhibitor resembles the enzyme's normal substrate, so it actually competes for the active site, right? So if you think about that, you're competing for something. So hey, you know, maybe this is the active site right here, and here comes my fist, but what if there's a tennis ball stuck in here? And all of a sudden, the normal substrate can't get in there. Well, that tennis ball would be a competitive inhibitor. It's competing with me for that same active site. So what can we do? Well, one way to overcome this is to increase the normal substrate. What if I had, you know, 20 tennis balls in this room bouncing around? Oh no, they might hit in here. Well, how can I overcome that? Or what would a medicine be or something like that? Would be, well, let's add 50 million of these, right? And if you add 50 million of these, well, now that you're more likely to have this reaction still happen. Tennis balls are basically obsolete at that point, right? So increasing the normal substrate concentration. And here you can just see a graphic of that. And when you go into non-competitive inhibitors, this is important for you to understand basically protein structure and function again, because you can bond something somewhere other than the active site. So here's normal, that's the way it should work, but oh, and by me bonding maybe something down here, okay, now the active site, just that little bit of bonding changed the active site, and now it's turned off. And it doesn't necessarily mean this is a bad thing. This is something you'll learn about if you go on to AP Bio, or if you take some more bio in college or Bio 2 honors. You can get into how this turns on and off. Sometimes is a good thing. It's like a switch. But anyway, the enzyme doesn't work here. Okay, and that's all because you're not competing for the active site this time. You're binding somewhere other than the active site, but it still changed the active site. Okay, so we get to feedback inhibition. All right, and this is actually really, really cool. I love reading about this stuff. So it occurs when products block a metabolic reaction pathway that otherwise creates something needed by the cell. So it's a little confusing at first. This is what we call a biosynthetic pathway, let's say. And our whole point is, okay, we need product D, okay, whatever this green star is here, that's what we're after. And to do that, we need to start with A, okay, and maybe A came from the environment. Maybe A is in our diet or something like that. And as long as we have A, well then enzyme 1 will convert A into B, and enzyme 2, which again, hopefully we have DNA through gene expression, we know how to make all these enzymes. Enzyme 2 will convert B into C. These are called intermediates, okay? And eventually enzyme 3 will make our product that we're after. But what if we have a ton of D, right? What if we don't need this green star anymore? Well, what can happen is, you can actually use D to inhibit enzyme 1. So now even if you have a whole bunch, maybe from your environment or your food or something, of the starting molecule, if you inhibit this enzyme, you just shut this down. And why would you do that? Well, if you think about it, if there's a whole lot of these green stars floating around, it makes sense that we wouldn't need any more, right? So that's, if it's weak bonding, sure, you can reverse it. Okay, and this is something that cells do all the time just so they don't waste materials, right? You shouldn't continue to make materials that are not currently in demand. What a waste that would be, right? It's like mowing your lawn over and over and over again without giving the grass time to grow. That's a great analogy right there. You can use that if you want to. So you look here, ATP, for example. ATP, man, you need a ton of ATP all the time. However, sometimes you make way too much. You need a lot more ATP if you're, in the middle of a soccer game or something like that, as compared to if you're sleeping at night, which you still need a bunch then. But so maybe you want to bond to something that makes ATP, which we'll learn about soon. Right? So it's very advantageous because it prevents wasting valuable resources in the cell. Okay. And finally, I decided to throw this in here. Just kind of cool how now we know some things about enzymes and we can get into how they work. So cyanide, it's a poison that inhibits ATP synthesis during cellular respiration. I have no idea how fishermen do this. I mean, I have the best fishing equipment and I can't catch anything. So I'm thinking maybe I should start using some cyanide to fish with, but I don't really get it. And I don't want to mistakenly, ingest any cyanide and lower my blood pressure and vasodilator. And no, thank you. But anyway, I can't catch fish ever, and I love fishing. So cyanide, I guess, can be used there, and you catch the fish that way. I don't know if I would suggest that. But you can see that it was also used as a suicide agent. Right? Espionage agents used to have those glasses, and they would chew on the back of them if they got caught, and the cyanide would kill them. So pretty crazy, pretty crazy. And if you can't make ATP, well, as you learned before, ATP kind of does everything for you. It keeps your energy, all your reactions, a lot of your reactions, almost all of them need ATP. So without that, you can't do your reactions and you die. Right? So that's one example of cyanide inhibits enzymes that help create ATP. Death is imminent. All right, sarin nerve gas. I remember, I was, I guess, just about to start high school or just starting high school when this happened, and boy, oh boy, that was terrible, right? Sarin nerve gas, what it does is it binds to an amino acid found in acetylcholinesterase. And by the way, a lot of enzymes, we talked about this before, end in ACE, right? ACE is an enzyme. So sucrose was the sugar, sucrase is the enzyme. Okay, cellulose, right, like plant cell walls, fiber. Cellulose is the sugar in that case. Cellulase would be the enzyme. Okay, so acetylcholinesterase, that's an enzyme, and it helps your nerve cells work. Basically, you send these chemicals, and they have to jump these gaps in your nervous system, so your signals can get from your brain to all over your body. And unfortunately, it didn't allow people to send those signals, so they basically got paralyzed and just died, which is terrible. So not all good stuff with these enzyme inhibitors, that's for sure. You can see here malathion and parathion, they act like sarin in insects. And a lot of people don't really like this stuff because, hey, look at that picture. I mean, we're spraying our food with this stuff, but theoretically, the small doses that we use aren't enough to hurt us, but the World Health Organization considers malathion. Yeah, they came out and said it's probably carcinogenic to humans. So, but we use such small amounts that hopefully we're okay with that. But I don't know. We are loading our environment and our bodies up with these chemicals. But anyway, in this case, we're using it for good, right? So farmers can have a lot of crops. We can kill a lot of insects that are troublesome pests, and we have food to eat because of that. Okay, but maybe organic, non-chemical is still the way to go. Yeah, definitely is. This is a crazy story. So many antibiotics, right, work via inhibition. So antibiotics are medicines that are antimicrobial. They kill bacteria, right? And this guy here, Alexander Fleming, it's nuts. He mistakenly found penicillin, kind of. Well, he was in his lab and he basically he put a little bit of his like nose snot, I guess, on a petri dish, and he forgot about it. And this was around World War One. He was Scottish, I believe, and he left it on his desk. He was in the army, and he felt terrible because he saw a whole bunch of soldiers. They were dying from not necessarily getting the wound, but the wound festering and the bacteria getting in there. And he was trying to tell people there's these little germs in there that are hurting people and killing the soldiers. So he was trying to figure out a way to stop this from happening. and he put a little snot on a petri dish, and he forgot about it and left this petri dish apparently on his desk for a couple weeks. And when he found it again, he realized that, oh wow, my snot prevented bacteria from growing. So nothing really grew around his snot on the dish. And actually what it was his snot had lysosome in it. Okay, and well, lysozyme, excuse me, the enzyme lysozyme, right? Your tears and bodily openings have this to protect us, right? We gotta have lysozyme. So any things that get inside of your eyes or wherever, you have to be able to shred it. You don't want these bacteria getting in and causing harm. So he actually found lysozyme, which is a very important enzyme. And he did another experiment then when he had a petri dish and he left it on an open window. And what happened was some mold landed on the petri dish and bacteria would not grow around the mold, he discovered. So he said, oh my gosh, there's something going on with the mold here. And it was in a penicillin family. So yeah, that's where penicillin came from. So because of his hard work, I mean, just imagine by World War II, you know, we have tons of people. You can see the poster here, a very famous poster. We had a lot of people, their lives were saved, all because of this doctor figuring out how bad certain bacteria are. A lot of bacteria are very healthy and good for us, but because of him, we know that, you know, probably a lot of us wouldn't be here, right? Because a lot of those troops were saved because of antibiotics. Amazing, absolutely amazing. Penicillin works by inhibiting an enzyme that bacteria use to build their peptidoglycan layer, their cell walls. And finally, you all know about ibuprofen probably, right? Like Advil and aspirin inhibit pain-inducing enzymes. Oh, okay. So if you're triggering a pain response in your body, your body's letting you know, ow, this hurts. There's something going on here. But a lot of times it's like, yeah, but guess what? I got school, right? I have work. Help me out here, body. I need to get through this. So what these drugs do is they kind of inhibit the enzymes telling you that this hurts. So it makes the pain go away a little bit. HIV drugs, there's lots of different HIV drugs, but a lot of them target the viral enzymes, right? It's really hard to beat a virus. And cancer medications, a lot of them work with enzymes for things that we're going to cover soon. Cell division, right? For your cells to go ahead and copy their DNA and divide, that takes lots of enzymes. So if you could find little chemicals that interfere with the enzymes that have your cells rip apart, that's a pretty good cancer therapy there. So anyway, that's a little bit about enzymes, and I hope you enjoyed it, and I hope that made sense. And again, feel free to shoot me some emails if I can clarify any of that. Okay. Thank you very much. Take care.